Assalamu alaikum and welcome back with the next part of this last section of your course. Another technique which is used in the recombinant DNA technology is the northern blotting. So northern blotting is basically blotting or joining together of RNA and DNA part of the life molecules. So DNA, we know it is transcribed from the RNA. DNA is transcribed to RNA. RNA is produced. Now we want to study. There was a time in history when there was no information, or yet the real-time PCR was not invented, which could tell us exactly how many RNA molecules of that particular genes are present in the system, in the blood, or in the cell. At that time, if we keep in mind the central dogma, that is DNA is transcribed to RNA, and then that RNA is translated into proteins. So these are two steps. First DNA to RNA, then RNA to protein, and then protein is basically going to express or going to show the phenotype. Now the first question, whether DNA is transcribed or not, or gene is transcribed or not. This is the first step. And the second step, whether that transcribed RNA is actually translated to protein or not. So to verify the first step, we use the northern blot. So normally RNA is single strand, and we convert it into the cDNA, complementary DNA. And complementary DNA means every U is converted to T, and it's now single stranded, it's more stable molecule as compared to the RNA. And this single strand, with the help of certain probes, which are that of given DNA, they are joined together and they complement to each other. And when they are joined together, those probes, they are going to eliminate the light. And those light are light signals, or those dye signals, they are red. And then we are able to identify whether the given given DNA is expressed or not, or given gene is expressed or not. So in first step, you get the RNA, you extract the RNA from the cell or any tissue source or from the blood, whatsoever. And then RNA is transcribed, and then with the help of the simple PCR, and then it is run on the gel. And after running on the agarose gel, then it is transferred with the help of the blotting onto the nitrocellulose membrane. And then here we can see it's the nitrocellulose membrane. And on the nitrocellulose membrane, in the next step, we add the probes or the labels DNA, which is normally having the phosphorus 32, and that is a radio label here. And this, when it is attached with the single strand of the RNA, then these joined together double strand, it will give us the signals, and those we can see on the X-ray film or any other source. Now the instruments are available, JetDoc systems are available, which can directly able to read this data without blotting them. Okay, the same method is explained again over here. How will you do it? The DNA, then it is cut into small fragments. This is called the basically DNA to DNA probing. The previous one was the DNA to RNA probing. And then cut the DNA in first step, and in second step, what we do, that DNA is run on the gel, and in the third, they are transferred to the nitrocellulose, and in the fourth steps of the experiment, this nitrocellulose transferred DNA is separated, and then in the fifth step, here we filter is exposed to basically radioactivity, because the probes are radioactive. As I mentioned previously, these are phosphorus 32, and then the part of the DNA which has joined together, that is indicated in the form of the uh, band over here. Alright, if we go to the details of the vector, so we have different part. The number one is the ori, or that is the origin of the replication, from where the replication starts. I have just told you in the last lecture that the vector is basically a circular DNA and is primarily originated, or it is retrieved from the bacteria. So they have origin. Origin mean that sequence that will help to transcribe the given DNA part which is inserted into this vector. So this is basically origin is a source of replication. Then the second important component is the selectable markers. So these could be antibodies, genes like ampicillin, or these could be in some vectors, especially which are related to the functional expression of the genes. So there are some GFP reporter genes or some other reporter genes which are expressed, and they help us to select whether the cloning has occurred or not. And then the third most important part are the restriction sites, or the cutting sites of the different enzymes, which help us to cut this fragment. And once it is cut, then we cut the DNA which we want to cleave with the same enzyme. For example, if we are using BAM-H1 for cutting the vector, we will be using same enzyme BAM-H1 for the cutting the DNA, and that DNA then it would be inserted into this vector with the help of the ligase enzyme. Okay, this diagram indicates another the more explicitly or more closely the different restriction sites. So here you can see the polylinkers, or these are the restriction sites. There is another genes, reporter genes basically added to that, and this is PUC19 vector, a commonly used T vector. So less Z plus is there. So less Z, they basically express the blue colors. When this gene expressed, the colonies on the agarose on the plate in the media, they will be producing the blue colors. And you can see all the polylinkers are the restriction enzyme. They are in between the less gene. So if the gene is cloned in between the less gene, then the expression of the less Z gene, it would be stopped, and there would be no blue colonies. And then here you can see MPR. So this is basically ampicillin resistance gene. So here three different ways or procedures are given over here for cloning DNA into the vector. The first one you cut with the same enzyme, and it produces the overhang in both the circular DNA and your desired DNA which you want to clone, and then you ligate it, and you get the results here. And the second procedure is slightly different, and here you produce the blunt end, and to get it to clone, here you have you can see you produce the overhang, but you add some other linkers, and with the help of the linkers you clone it to the vector. And the third one is also similar to this one. So there are these three different type of approaches for the cloning of the desired DNA in the vector. Okay, once your gene is cloned, then selectable markers, the less Z example is given to you, and if the less Z selectable markers, they could be resistant antibiotics, or if that circular DNA is transferred to the bacteria, then it will produce the resistance gene. So if you add that antibiotic to the media, the cell will survive and colony will be formed. So this is the marker 1. The second marker is the reporter gene, that is basically less 3 gene, less Z gene, I just told you. And it will produce basically, if the desired fragment is cloned in between the less Z gene, then you will get the white colonies. And if desired is not cloned, then you will be getting the blue colonies, just shown in this diagram, in this plate on the media. Now these different plasmids, so there are different type of plasmids. The number one, we call them simply plasmids, or these vectors are different types plasmids. So they can clone up to the 15 kb DNA fragment, and we use the transformation procedure to enter into the bacteria. The second type is the phage lambda, and they can take up to 23 kb, but we use the infection of the, because phage is basically a, phage lambda is a virus and it is infected to the bacteria, and then we can transfer our DNA. And the third one are the cosmets. So they have ability to take maximum size of the DNA up to 44 kb, and generally they are used to produce the genome libraries, genomics libraries. So again, this is a virus, and we use the infection method to introduce this DNA to the bacteria. Thank you very much.